Tihei Māori ora. He mihi nui ki te mana whenua o tēnei rohi. Waitaha kate mamo i kaitahu. Ki na mauna whakahi, ki na wai tiri nei, ki na rangatira huhua, tēnei te mihi, tēnei te mihi. Ki na mate o te wā, rātou kuia, whetu rangatia, haere, haere, moi mai rā. Rātou kia rātou, tātou kia tātou. I nā rangatira, i nā ahorangi, i nā kaimahi, i nā tauira, i nā tangata katoa, tēnā koutou. Ahorangi Craig Bunt, tēnā koe. He mihi hoki tēnei ki tō whānau me o hoa. He rā nui tēnei, he kaupapa nui hoki. Nō reira, tēnā koutou, tēnā koutou, tēnā tātou katoa. Greetings to you all. I am Tony Ballantyne. I'm the Deputy Vice-Chancellor External Engagement for the University, and I have the honour of hosting this very special occasion this evening. A very warm welcome to all of you who are gathered with us in person here in the Castle Lecture Theatre, and also a warm welcome to those of you online, both in the present and perhaps in the future, catching up on the video, including greetings to Craig's parents who are hoping are online live, but maybe catching up on it in the future if that hasn't quite worked out. So it's great to see you all. It's great to see you all because you're here on what is a very special occasion. The fact that some of us at least are wearing these robes underlines that it's not a usual day in the university's life. The robes are an indicator that this is a special moment. We wear these robes rarely these days. The two main occasions are at graduations, when we celebrate our students completing their academic journeys with us, and at these inaugural professorial lectures, where we celebrate our colleagues attaining the highest academic rank in our institution. So it is a fantastic occasion to celebrate the work, the teaching, the research, the career of Professor Bunt. These are special because they are a celebration of the core business of the university. They embody and celebrate the endeavours that are at the heart of the life of our institution, the scholarly journey, the creation of new knowledge, and the transmission of that knowledge to students, to colleagues, and to the interested public. So it is great that we have all gathered here this evening to celebrate both the outstanding scholar that Professor Bunt is, and more broadly, to celebrate the importance of knowledge in our community. And of course, the timing of this gathering is auspicious with the rising of Puaka Matariki. It is a great opportunity to look back and to take stock. And if any of you heard Professor Rangi Matamua on the radio this morning, he said Matariki is about three things: reflecting on the past year, and more generally reflecting on the past and how you've got to this point, taking stock where you are now, and also looking forward. And that seems to be very much in line with the kaupapa of an inaugural professorial lecture. So Kati, I think that is enough from me. I will now hand over to Professor Phil Bremer, who's our acting DVC academic, who will provide a full introduction of Professor Bunt. So, nō reira, tēnā koutou, tēnā tātou katoa.
Thank you, Professor Ballantyne. Tēnā koutou katoa. It's my great pleasure to introduce Professor Craig Bunt. Craig is a proud alumnus of the University of Otago. He completed a Bachelor of Pharmacy degree of honours in 1990 and a PhD in 1996. Craig's PhD was on factors influencing the microencapsulation of bacteria, and his co-supervisors, Dr. David Jones and Professor Ian Tucker, will be well known to many of us here tonight. After completing his PhD, Craig worked first as a research officer with InterAg, a veterinary pharmaceutical company, before accepting a role as a senior lecturer in pharmaceutics at the University of Auckland. This role was then followed by a spell as a senior lecturer, a senior research scientist at Ag Research, before he returned to academia as a senior lecturer in animal sciences at Lincoln University in 2015. In 2021, Craig had the honour of being appointed the inaugural Professor of Agriculture Innovation at the University of Otago, a position in which he has excelled. Craig's research focuses on the delivery of bioactive compounds, and his scientific contributions span veterinary pharmaceutics, food science, and environmental management. As you will hear from Craig this evening, his research has led to the development of unique transformational technologies which have helped to promote environmental sustainability and led to improved agricultural practices. Craig is the author of 60 peer-reviewed research publications. He has given over 50 oral presentations, and he has 16 patents, including 6 granted in the US. Craig is on a number of scientific and industry advisory boards, and he's been awarded many honours and distinctions, including an OECD Fellowship in 2015, 3 awards associated with the Ideas on Entrepreneurship Challenge in 2011. He was the Waikato Agribusiness Innovation Award Supreme Winner in 2003, and in that same year, he had a paper recognised by the Controlled Release Societies for an outstanding paper. At the University of Otago, Craig leads the AgVet Controlled Release Lab, which researches agri and veterinary controlled release formulations. Craig is also a member of the University of Otago's Food Waste Innovation Team, which measures food waste, develops reduction strategies, applies innovative technologies, and works to modify producer and consumer behaviour. This evening, Craig will reflect on his career in agriculture and pharmaceutical sciences and discuss some of the work he has carried out on controlled release formulations for agricultural and veterinary applications, with a particular focus on the development of advanced technologies. Please join with me in welcoming Craig to the podium.
Tēnā koutou. Ko Rāhotia te monga, ko Kura te au te awa, ko te ate awa tōku iwi, ko Waikawa tōku marae. No Greymouth ahau, ko Robert, roa ko Lynette Mātua, ko Jill, tōku wahine, ko Caswin, roa ko Emily, nā tamariki. No reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you for coming out on what's technically a Friday. So I very much appreciate it. Thank you. How do I go from farm, PH, to FARM? I'm a bad speller. Those of us who remember School Cert, in my defence, I got 51 for School Cert English. Somebody still corrects my grammar to this day. So I arrived here somehow in 1988, second year of university. I spent a year at Canterbury. A couple of years before that at high school, I'd been to a careers night and heard about pharmacy, that you could go to university and get a degree in pharmacy. And I wanted to do pharmaceutics. That sounded interesting. So I started on a career in pharmaceutics. I've blanked out the names to protect the innocent, but we are in alphabetical order. That doesn't help trying to figure out where I am. Some of us may recognise Lloyd. He's still practising around Dunedin. Hasn't really changed much, just the hairstyle. I have to thank Justin. Now, where's Justin? Towards the end of my first year in a class being taught by Professor Mike Roberts, he said, I have a summer studentship. If anybody would like to apply, you can spend some weeks in the lab working on a research project. And I had no idea what a summer studentship was. And Justin was sitting next to me and he just nudged and said, we should ask about this. So we went up and asked, and we were the only ones who asked, and we got the positions. And that's where it started. So I got to spend a wonderful summer in the lab, experiencing research outside of the teaching environment, talking with postdocs and PhD students, where up to this point I had no idea who they were. And I got to see my lecturers and Professor Roberts in a totally different environment, sitting down in the staff room talking about science, not an assignment that's due next week or a test or something. So thank you, Justin. Just right place, right time. That's me, by the way. Just to help. When I had hair and curls. So I have to thank Professor Mike Roberts wholeheartedly, who set me down this pathway. Mike, in the world of pharmaceutics, is an early trailblazer in the field of transdermal drug delivery. He's internationally well known, immensely respected. And if you've ever met Mike in person, it's no accident that his photo is larger than the rest. He is an enormous man, but the most gentle, kind, encouraging man you will ever meet. His advice while a student and years later when I'd bump into him at a conference on where you could take your career and how to approach what you're doing is just absolutely outstanding. I loved introducing my grad students to him whenever I could, because I knew that they would get something out of talking with Mike, because he always had so much to give. David Jones was new to Otago, fresh out of Queen's University Belfast. And what I'd done in the lab that a previous summer with Mike grew into an honours project with David. I didn't know it at the time. I thought that I and some colleagues were keeping him up late at night helping us write our dissertations. It turns out he's an insomniac, and it was no small. It wasn't really, we weren't putting him out. But he sat there with us and he walked us through the entire process. And he'd be there in the lab with us and he'd help us talk about and figure out how to solve problems or approach things. And so from that wonderful engagement in an honours project, I thought, I don't want to leave this. So I didn't go out and register as a pharmacist. I wanted to stay in the lab, be very much a pharmaceutical scientist. But I've benefited from having had the clinical and practice training I had to face while also looking at pharmaceutics. It's not just what a tablet looks like and how it functions. It's what's the patient thinking about it? How do they face it and interpret it? And what's this medication going to mean to that patient in terms of their quality of life? Now you can apply that to humans, but equally to animals and equally to the environment or a plant. So with David, I went into a PhD with Professor Tucker, who was newly arrived as well from Queensland. And David went off and had a conversation with Frank. And thank you, Frank, because David came back and said, right, we're going to do something in vaccines. There's challenges and issues around vaccines, how well they work, and how often we have to give them to livestock. Could we actually find a better way to do it? And so that's the PhD topic. I started looking at vaccines. Is there a better way to formulate them? Could they work better? Could we get rid of having to give multi-shots? We were asking those questions back then. It's very relevant today when we think we're up at our third, fourth booster for COVID. Wouldn't it be great if we could just have one shot? There's been no real progress in being able to achieve that, but that hasn't held back other discoveries around vaccines. And so from 91 to 95, I worked on looking at how I could put small bacteria into tiny particles in the worst possible way you could try to do it. We didn't make our life easy. We thought if we could put them into a little particle that was a biodegradable polymer that had function and form and other properties, we could gain some benefit. But the way to do that needed harsh chemicals and harsh mixing. Could we somehow protect the bacteria or find the right sweet spot of conditions and get the thing to function? Could the bacteria survive the process? There's far easier processes we could have used, but they wouldn't have been able to deliver the performance we were hoping to see with the sort of material we were working with. There's a scene in the movie Awakenings where Robin Williams is being interviewed for a job and it's commented that he tried to extract something from 4 tons of earthworms. And the interviewee says to Robin Williams' character, but that can't be done. And Robin Williams says, yes, but I know that now. And I think that movie must have come out around the time I was doing my PhD or soon after. And that scene has stuck with me. And I thought, had I remembered it right? So I looked it up recently and rewatched it on YouTube, and yes, I remember it exactly. I know that now. I know now what not to do when trying to get bacteria into these little particles we constructed. We wanted this shell arrangement and these little wee pockets here in which we would put bacteria. And we thought we came up with a system where we could control how things stuck to different materials. That's what the graph on the far side is trying to show. And we thought we had a wonderful way of doing it. But every time I put bacteria in there, they died. I had an assay that could quantify they were dead. There was definitely nothing alive there. So I always said, I now know what not to do. My PhD, I was in the lab from 91 to 95, but I had to wait another year to graduate. Students used to protest a lot more vigorously back then than today. And I couldn't graduate for quite some time because students kept storming the council. And every time they were going to graduate a cohort of students, the meeting had to be disrupted and it kept getting put off. And I was getting frustrated. Oh, I'm not graduating yet, I'm not graduating yet. Can't get into 1997. Could the council just meet and please not tell the students where they're doing it? So eventually I graduated. So my CV, if I look at dates and stuff like that, it looks like I took 6 years, but I didn't. Still to this day in my office, if I hear a protest outside, my first thought is it's the students, but lately it's more likely colleagues. So after my PhD, I went straight into an industry job. I was very lucky. Somebody who had been at University of Otago had recently left to head up a team for a company in Hamilton called Interag. Interag is a rather unfortunate name. We have this habit in New Zealand of anything ag-related tagging the word ag onto it at the start or at the end. But you travel through parts of Europe and they think you're from Inter, because companies are often having the name ag after them. It's their equivalent of limited or something like that. So if I was in Germany, I was always from Inter. So I was at Interag for about a decade. And the team I joined was a small team with quite a specific task. And we were looking at this product here that's used to time, to control when a cow will go into heat. So you can time when you inseminate it, and you can then time when calving is, so many months later, and you can time all that up with our dairy cycle, our grass-fed dairy system. So I was on the team that looked at taking the version that was being marketed mostly around Europe, parts of Asia and Australasia, and formulating that in a way that would be more acceptable for the regulatory requirements of the US. We also looked at other versions for sheep, biodegradable polymers as well. This one's silicone, another biodegradable one, and then one for pigs, the difference being anatomy and size of the animal. And so the colleague or the friend, the boss, the mentor who had left Otago about a year before I'd finished my PhD was Michael Rathbone. And unfortunately, Mike passed away last year. And what we did at that company, we could not have done without Mike. He was somebody who went from academia to industry and absolutely flourished. He could bring people together from any discipline. He could bring people in a room together and have a productive meeting and everybody get them on the same page and everybody heading off in the same direction. He would edit books prolifically, and it was a way, to me it illustrates just how well he got on with people. Very few people ever declined the opportunity to contribute to a book Mike would be editing. This is just 2 of something like 8 he edited over his career, ranging from human oral mucosal, so mouth drug delivery, to veterinary. And the people that contributed chapters to these books were leaders in industry, leaders in laboratories around the world. They did not hesitate to engage with Mike. And I say, again, what we did there would not have happened without Mike. And sadly, he passed away last year. So what we did was, we took an existing product, stylized here, think of it sort of like an IUD, but for cattle, and it goes into the vagina. And then while it's in there, it releases progesterone, a natural hormone, at a controlled rate, and that influences the reproductive cycle. The version we started working on had been developed by scientists at DSIR and a company called AHI Plastics. And they developed it for many markets, but they needed a slightly different version for the US market. And so the research we undertook took it from still essentially a T-bar thing, but to something that was slightly different. And this paper actually describes the research and the stages we went through to get from this to this. We did a lot of typical lab work, formulation work, and what we usually do in formulation science, change the ratio of ingredients and how we process them and prepare them, very much like cooking. What happens if you adjust this ingredient, influence that one, overmix it, undermix it, what do you get? And so all of these graphs are showing with the slopes is that if we change something, we get a change, and we can understand what that change means if we apply formulation science. So my colleagues in formulation science will know that if we increase drug load, we probably increase how much drug comes out. If we change the material properties of the bulk of the material, we'll probably change how things come out as well. So we did the usual, increase how much you have in there, you'll increase how much comes out. You make it a harder material, it'll take longer to come out. That's the beauty of test tube work. It's easy to do, it's there in front of you, it's usually predictable, and then you go into the animal. And so the figure on the left, a flat line, is showing us nothing happened. We changed things along the bottom axes, but this flat line here, everything we'd seen in the lab didn't happen in the animal. Turns out that wasn't a disaster. With what we knew from how it worked in the lab, and then started to figure out, well, why isn't it working like that in the animal? We were able to make something that functioned exactly the same as the earlier version, the older version, and make a newer version for the US market that had less drug in it, but still was able to obtain the same delivery. So less in, same response. That kind of goes against the understanding of models of how drug usually gets out. And so we realized we were working with something different than expected, novel and non-obvious. So that actually allowed us to get a series of patents around that. I was lecturing on these patents today, actually, to a class of agri students, explaining what a patent is. So we had a period of exclusivity where only we could make this product here. And more specifically, we had a product that was ideal for the US market. And so we took that through to US registration, so approval to sell it in the US. And that was right at the turn of the 20th century. And so up until that point, this type of product wasn't available in the US. But it's kind of interesting when you see a letter from the FDA on their website saying, seize any packages on the Mexican or Canadian border with the word Hamilton and Interag on the packaging. So our products were on the market in Mexico and Canada, but the US FDA said, no, they can't come across the border because it's not approved here. You have to seize them, seize and destroy. But we were getting our product prior to approval into the US because a lot of US researchers at universities were using this to understand bovine reproduction. And so it wasn't unusual to have a visit all the way down to New Zealand by a US researcher who might be going on to Australia or Japan or somewhere, visit us in Hamilton and go home with a suitcase full of these things. Because they weren't importing them and they were bringing them back to a university for a research purpose, they were allowed to bring them in, they weren't destroyed at the airport. What it meant was also that we had a lot of US researchers who were very familiar with this product, knew how it worked, actually showed us how it worked in the North American market or North American farming system. And that helped with registration and it also helped build a market before it got there. And so this product here is New Zealand's first researched and FDA registered pharmaceutical, human or veterinary. And in terms of the quality of manufacture, human or veterinary is no different. I joke, if there's a zombie apocalypse, don't head to the pharmacy where everybody else is breaking in for the medication. Go to the veterinary practice. There's far more and the quality is just the same. And so taking that through to US registration got the attention of some very large companies, and we ultimately ended up becoming what's called a toll manufacturer. Pfizer, Pharmacia, they were all merging at the time, took on that product, sole distribution rights, and we continue to manufacture that in Hamilton. To this day, it's manufactured in Hamilton and exported all around the world for Pfizer. We were also had an agreement with Pfizer to continue researching new products, new ways of doing these sorts of things with veterinary pharmaceuticals. So where previously we'd found that we changed things in the lab and we put it in the animal and it made no difference, we went back to the drawing board and we thought, well, is there a way we could actually still influence things, that what changes in the lab still does actually show a difference in the animal? And so we found a way that we could change the formulation, put it in the animal, and we'd get a change in blood level. The thing would actually work as expected. Normally, these sorts of products, you're only aiming for this blood level that this graph is representing down the bottom here. Just this amount is fine. But we found we could double that, 5 times that. That higher level would have enabled endocrinologists and animal reproduction scientists to try a whole lot of different things or different ways of treating animal fertility. But sometimes you find a way to do something better, but then you get asked, yeah, but can you still make it just work exactly the same? Better isn't always wanted or needed. Being better and being different can cause all sorts of problems when it comes to registration. If you want to register something new that's a pharmaceutical that's based on a previous type of product, the closest you can stay to that previous product can just help in terms of registration, unless there's a dramatic improvement or better way. So we ended up, while knowing we could, making something that worked exactly the same as the previous product. But this one, unlike the previous product, is totally biodegradable. It will undergo a breakdown in the environment, microbes will eat it, and there'll be no trace left. The earlier device is made from silicone. It won't break down in the environment, it won't create microplastics, but it will be dug up by archaeologists in about 2,000 years wondering what these things are. When you're in industry, you keep a close eye on the competition. This is a device produced by a company, again in Hamilton, that contained some complex electronics, a way to retain in the animal, and little reservoirs and valves and solenoids and things to pump out one drug and pump out. This could actually give a whole therapeutic regime of continuous drug and a shot here and a shot there over a week. And so you could put this in theory and then come back 8 days later and the animal's at a certain stage in the reproductive cycle. That was the theory. It actually got to market, which is great because then you can go out and buy these things if you're the competition. You don't have to wait for one to fall off the back of a truck. So we went out and bought a whole lot of these, started testing them. Maybe within about 2 weeks knew more about this than the company knew. About 2 weeks later, they actually recalled all of their stuff from the market. It just did not work. They claimed it was because of this retention device, the way it stuck in the animal. We found that was about the only bit that did work. These complex electronics, totally covered in basically Vaseline to protect them, just couldn't survive the environment they were going into. The whole thing just failed and it never functioned. And we thought that this is actually over-engineering to try and address a complex problem. So we thought, how simple can we make it? How much can you pare it back? We thought, well, there was a technology we were aware of that produced gas in a syringe to put lubricant into industrial equipment so that you don't have to go along every day and squirt with the grease gun. And so we started playing around with that, and we came up with a syringe that has a tiny little gas generating unit at the bottom there, and you can set it to go off at a rate that takes hours, days, or weeks to just basically push a syringe out, then push on other pistons and give a dose of this and a continuous something else. So we were able to show that this device could give a continuous delivery of something and also give a couple of pulses of something else. So you could start to build in a whole treatment into the one device. The device goes in, you don't have to keep visiting the animal, yarding the animal, and treat it from a very simple thing. This, while it's a simple device, it was incredibly expensive to do. It's injection molding. You have to build a mold or have a mold machined going into a complex plastics making machine to make these components. So just the equipment well, not even the equipment, the mold to make this was like about $40,000, and going in a $200,000, $500,000 plastics machine to make something that ultimately probably would have, the ingredients, the plastic, we would have wanted to have kept the cost quite low. So we sort of parked that one. It was probably functioning great. It probably could have been better. It could have been advanced further, but it really wasn't going to replace how things were used. But right around about that time, I met a student at Waikato who was interested in doing a master's project. And he had previously been an electronics engineer for the New Zealand Air Force. And so he knew electronics, and we'd gained all this experience from these devices in animals. And so we had a syringe that's got a gas generator pushing this piston. The good thing about having the electronics inside the gas generating area is, it's always a positive pressure. Stuff isn't going to leak into there like the commercial one we'd previously seen. Having come from the New Zealand Air Force, where there isn't really much of a budget for anything, every time Peter said, "Oh, can I put this on it?" I said, "Yeah, do that." "What if I put this on it?" "Yeah, do that." So what started out as a project just to control how this syringe works, we also had a radio transmitter on it, putting out a signal every 15 minutes and asking if there was any other devices there. We had a motion sensor on there, and we had a light sensor on there. Peter said, "Why a light sensor? It's going inside the animal." I said, "If it detects light, we know it's fallen out." So we just thought, what can we do? And this was off-the-shelf components. But we could start to measure how active the animal is. You can time that up with the time of the day and know if it's sleeping or if it's moving more during the day than at any other time of the month. And you can start to then understand what behavior the animal is showing. You can understand what stage of the reproductive cycle it might be, or if it's under some sort of stress. So we also had this tied up and linked in with one of the very early robot milkers based up there as well, where you could actually have a closed system where the robot milker is taking the data off these things and understanding or getting an interpretation of what's happening with the animal, and then deciding whether this device does anything or not. It was a good idea at the time. It was never going to be cost effective. We were buying components off the shelf. Each one of these were $50 or $60. But a lot of what this does, you can now do in an air tag with a little solar panel. You can detect movement of the animal. You can use that to interpret the behavior, the condition of the animal, ID it, who's it interacting with quite cheap, highly functional electronics. And that's about when I finished my time at Interag and went on to other things. And my time at Interag was very linear. We went on from this and this and this. And it's quite encapsulated. Then for my next three positions, I thought, are they linear? They're not. They often had bits overlapping. They had projects started here, ended up being finished there. So I thought I'd bring them all together. If my PhD was, I now know what not to do, then my time at Auckland University was very much featured by welcome to PBRF. Now, PBRF is a thing where every academic in the country every 6 years or so gets assessed individually, gets graded. And so I walked into a PBRF round having never been in a PBRF round before. And my colleagues were like, "Oh, you haven't been at university, you haven't been in academia. You're going to struggle to get a good score." And the PBRF is around measuring the impact of your research. And I thought, no, that shouldn't mean that just because I've been in academia, been in industry, my research should be any less impactful or have any less impact. And so I thought, no, you don't have to be in academia for your research to be of worth, to be of impact. So I put all the stuff I'd done in industry into my evidence and got an assessment, and it was a good assessment. I had arguments with the research office who were saying, no, a paper is not the same as a patent. A paper is worth far more from a journal than a patent is. And I was going, but that patent was licensed to Pfizer. Doesn't that show impact? So I felt like I had to educate the research office. Where in the guidelines does it say a paper is worth more than a patent? We got down to arguing. They sent me the list and they said, here's the list, papers are at the top. And I said, nowhere does it say that this list is a list of priorities. So I have a passion to this day about PBRF and understanding exactly what it is, which probably explains why I'm on a tech sector reference group that's trying to reframe and redesign the PBRF system. And I think the PBRF system is a good system. I might get run out of the university by my colleagues for saying that. But it actually helps you understand why you're doing what you're doing. I then went to ag research for a bit. And this is where threads start to come together. I went into ag research, which was very much a corporate type model, but not as I knew it. When I'd worked in industry at InterAg, we had a CEO that trusted us and said, you're the experts, you do this. The CEO we think is a myth. They do exist. Bill Thompson very much put a team together and said, I've hired you because you're the expert, you do it. And that's why Mike was able to flourish and show what he could achieve. And he had a team around here that could do that with him. At Ag Research, that's not the situation. If the funding model for universities is being discussed and how it fails university systems, then and the CRI, Crown Research Institutes, the funding model for that is absolutely atrocious and devastating for science and the people who work there. And I can say that now because I have academic freedom, but I could have never have said that when I was at Ag Research. And we'll get to another point about that. And then I literally moved across the road from Ag Research to Lincoln University, into the middle of a PBRF round again, having spent a significant proportion beforehand not in academia, and a new PBRF office at an institute where I had to re-educate to them why commercial research is the same in terms of impact as a publication in a journal somewhere. What I went to look at Ag Research though, and this is where threads start to come together, was can we use microbes as an alternative to agrochemicals for pasture pest control to help plants grow better, as herbicides and such. So rather than chemicals, use the natural disease-causing organism or the beneficial microbe that helps a plant grow. Plants can't grow without microbes. When people say soils in south of Auckland are sterile and nothing grows there, and it's like there's market gardens. You just cannot have a plant grow if there isn't a microbe or the right microbes there in the soil. This is grass grub. It's a major pasture pest in New Zealand, and it can be infected by this bacteria growing on a petri dish here called Serratia, and it causes a condition called amber disease. So this black line through the grass grub here is its gut. You can see its gut contents. When it gets infected by Serratia, the Serratia stops the grass grub eating, and the grass grub will die and create a little pool of nutrients for the Serratia to grow into. So the Serratia bacteria is a predator of grass grub. And it's a natural microbe in the soil. These two exist or can't exist. Well, the Serratia can't exist without the grass grub. But sometimes grass grub populations get out of hand, or soils sometimes are missing that grass grub. You might have had a soil that's not had a grass grub in there for a long time, so the Serratia doesn't hang around because it's got nothing to eat. Grass grub gets back in and gets out of hand. A very early product to take this from the lab or from manufacturing through to the paddock was basically injecting a liquid preparation, a liquid culture that needed to be refrigerated. It was full of problems. Had to be diluted with water. If the farm's water was on town supply and it was chlorinated, that killed it. This product didn't work in the market, partly because we didn't know exactly how to use these as alternatives to agrochemicals. Agrochemicals are kind of robust and idiot-proof. You can treat them in a shocking way and they'll still work when you put them out there. Put this in the back of your truck during a nor'wester in Canterbury for the weekend, and by Monday morning it's all dead. And you put it out there and you say, oh, it didn't work. Of course it didn't work, you didn't treat it right. So when I was at Ag Research, we were taking these microbes, putting them onto surfaces, onto zeolite, which is a mineral, into granules, and stabilizing them, keeping them alive and happy so that they would be on the shelf, be more robust than a liquid culture, and you could put them out there in the paddock and deliver this microbe as an alternative to chemical pesticides. And while from my PhD I knew what not to do, it didn't necessarily mean I knew what to do. Why a microbe dies and doesn't reanimate, why what looks like a perfect environment for it to survive yet it doesn't, is incredibly difficult to get our heads around. We can perform an autopsy on an animal, but how do you actually figure out how bacteria died? So we would typically do a lot of screening, and what we're looking for here is just anything that survives for long enough. And what we're looking at here is trichoderma, which I call a yeast, but my colleagues who understand trichoderma tell me off for that, but it's a fungi that resides in soil, helps plants grow, help them survive their environment. They can impart resistance to fungal attack. They can help plants access nutrients in the soil. And so if you have that microbe, that trichoderma, on the seed, before you plant it, then that seed will germinate in an environment that has these beneficial microbes around it. But it's no easy task to make sure that that microbe is alive during storage of the seed and planting and germination. So it's the same sort of thing we have with rhizobia and clover. The right microbe and legumes have those two together, and the legume, the bean, grows and it's able to capture nitrogen out of the air. Well, if you grow a legume and that rhizobia, that microbe that helps it do that, isn't there, then your legume won't grow. So we use these microbes across agriculture. But the challenge is getting them to survive. This is a bit of work with PhD student Scott, who was at Canterbury at the time, and he was in our lab at Ag Research. And sometimes we get a result. This flat line means that we could keep this Pseudomonas ADP, this bacteria, alive at room temperature, sitting on the shelf for 10 weeks, if we treated it this way. But if we did it another way, it's dead in 2 weeks. I started to think maybe it's how moisture is controlled. The materials we were using there, one was a New Zealand, what's called zeolite, a clay mineral. One was an Australian clay mineral. They're both the same class of mineral. One worked, one didn't. And so I was at Ag Research for about 4 years, putting microbes onto things, stabilizing probiotics for animal health, and also using microbes as herbicides for controlling thistle and things like that. Scott, after his PhD, went off to the US, and he went to the USDA working on formulations for wildlife control. So these are ways for controlling pest animals or improving the survival and health of endangered animals. I visited him at one point around about 2014, I think, and where he was at in Fort Collins in Colorado, there happened to be a herd of bison. And these bison are part of a herd that's been separated off from the bison herd that's in Yellowstone, which is about the only one in the US of pure bison. So it's kind of risky. The only place in the US where there are still bison is in Yellowstone, which is sitting on a volcano. So there's an effort to create a second herd of bison somewhere else in the US, and so they're looking at creating one in northern Colorado, Wyoming. And so these are some of the first bison cows that were brought in to start to build up a new herd. I happened to be there at the time when they were synchronizing them for insemination and embryo transfer. And so these bison here actually have a cedar in them, this thing I worked on back in my industry days. I go to this site and I see that they're actually using something we use here in New Zealand for in our dairy industry. They're using it to increase the population of an endangered species. I was chuffed to see that. I kept talking with Scott, and the next year I got an OECD fellowship, and I went back to his lab for 3 months. So we had a sabbatical in Fort Collins, and I happened to get back just as they were calving. It's kind of nice to see the year before something you've worked on is helping bison get in calf, and then you come back in the next year, and you happen to be there when the bison start calving, And you arrive at work one morning and the news cameras are all lined up because that was big news. That was one of the first pure bison to be birthed in that part of the US in a very long time. So while I was there, we were also talking with other scientists at the USDA, the Department of Agriculture there, about ways of doing things differently. And an issue with the bison is they are susceptible to a disease called brucellosis. And in Yellowstone, they require vaccination. But we can't vaccinate them with traditional darts because that would be leaving bits of plastic and needle lying around a national park. So we started talking about, well, could we use 3D printing to print a dart that we load with a vaccine? It's from a biodegradable material, so if you miss, it's going to break down in the environment. But if you get it into the animal, there's not a bit of metal getting into it as well. So this became a project with some people there, but also Ali Safodin at AUT, and a couple of PhD students worked on that. So I always joked when I had a 3D printer and people would ask, are you printing a gun? Because that seemed to be the standard question when 3D printers came out. And my answer was, no, I'm printing the bullets. Well, with what I started to understand about how we can stabilize bacteria better and more importantly, treat them better, we started applying that to food science as well. So Min was a student at Lincoln, where we figured out how to make a muesli where these flat lines at the top a probiotic bacteria on a dry muesli that doesn't require refrigeration. So you can open up your muesli, pour it in your bowl, and there's the probiotic already. You don't need a probiotic yogurt out of the refrigerator to do that. You might not like or be able to take dairy products, so we could put probiotics straight there onto that and stabilize it. It was a good bit of work that Min did, and she also did a whole range of taste testings of all of these types of products as well. Most importantly, we could put it on there and people couldn't tell it was on there. Probiotics only work if you're taking live bacteria. Don will recognize this. I mentioned before about academic freedom. When I was at Ag Research, I looked at a product that was on the shelf for animal health, and it was calling itself a probiotic. And I looked at it in the lab, and there was nothing alive in there. And somebody at Ag Research, when I commented to them about that, said, well, you can't tell anybody about that. And I said, what do you mean? He said, well, they're our strains of probiotic bacteria they're using. You can't tell people that they're now putting a product out with nothing alive in it. So when I got to Lincoln University, and colleague Malik Hussain there started talking about, I've been engaging with this company called BioBrew, who want to know what their product's like compared to others on the market. I said, well, I know the one that won't have anything in it. And so we did some work. So basically, the green dots are live bacteria, the red dots are dead bacteria, and this is just after 2 hours, half of them are dead. That's when you just leave a probiotic sitting out there and exposed to the environment. These are incredibly fragile bacteria. So we looked at 4 products sold on the New Zealand market as probiotics for animal health. These 3 here have less microbes in them than Dunedin tap water. There's practically nothing in there. Only one product actually contained large amounts of live probiotic bacteria. And I remember at a conference once I presented this data, and somebody from company B, C, or D came up to me afterwards and said, oh, I see you might have a problem with our products. I just simply said, I know exactly what they aren't. There's no legal definition of probiotic, so there's certain key things you have to keep an eye out for. Does it state how many live microbes are in there? And the more the better. And the giveaway is, if they say at the time of manufacture, they only know what it was like when it was back in the factory. Just want to wrap up with a comment about some work, more recent work. What happens when a material scientist like myself is married to a historian who takes a material cultural approach to history and mentions that early explorers in Antarctica were using an innovative, a scientifically advanced thing back in the late 1800s, early 1900s when exploring Antarctica, taking with them dog biscuits. We just think dog biscuits. Dog biscuits were a scientific advance of the age. And I just said, well, I wouldn't have trusted anything the Victorians made. And my wife, who's the historian, Jill Haley, said, well, how would we know? I said, well, we could analyse it. But we can't destroy this. This is a historic artefact, even though this is a broken biscuit in the collection of Canterbury Museum. I said, well, no, we don't have to destroy it. We can use instruments here at Otago University, or go down to, here's a good excuse for us to go down to Dunedin. We love visiting Dunedin, and working with Keith Gordon's group and Sarah and co, and Jeremy, aiming some spectroscopy at bits of the dog biscuit and getting an understanding what did they make these out of. And yeah, things that were looking like bits of bone were bits of bone. Things that were bits of brown were probably bits of meat. Think a cabin bread with a hint of meat in it. It was still a scientific advance of the age, and it was probably better than anything else they were still feeding. Thinking, fresh meat is still 70% water. And so we published this, and this illustrates an issue you can have when you've got a multidisciplinary team. We've got a historian, a materials scientist, spectroscopist, somebody understanding animal science, animal nutrition, writing a paper. We were lucky that we had an editor who could understand what we were trying to do. The reviewers couldn't. And many of you will know what I mean by we ran into problems with reviewer 2, who wanted all that boring history stuff out, put into the appendix. But no, this was very much a history paper using science to just simply support what the history was saying, or how we were understanding the history. We placated them by putting all the spectroscopy data and more into the appendix. And there was a lot of stress while writing this paper because the husband and wife don't always work together. But this is Georgia, and Georgia has a fetish for paper. She's a husky retriever cross. They eat paper for some reason. And one day I had to say to Jill, my wife, I'm sorry, the dog ate my homework. I was working on a printed out draft, and I turned around and it was shredded, and she had got to it. I'll wind up, and thank you so much. I'd like to thank the PhD students I've worked with, the postdocs, the masters, colleagues, friends from everywhere. This list is far too short. That there are so many people I am sorry I've forgotten. I've had an amazing time since starting here at Otago, moving off around the country and coming back. And normally I would have probably have ended my talk here, because I came here in the middle of a pandemic. Don't get involved in a new program at a university during the middle of a pandemic. But I can tell you something about what we've done in the last 2 years to wrap up. So the 2 years I've had with the program have been an absolute blast. There's a lot of questions of why is Otago doing stuff in ag? That's Lincoln and Massey. Well, if you're going to be on a farm or visiting a farm every day, then probably Lincoln and Massey. If you're somewhere else in the massive sector that is agriculture in our primary industry, then an Otago degree with some ag in there is probably going to ag-proof your degree. So we're engaging with farmers up in Ranfurly who are trying to get on top of pasture pests and put in mixed species pastures and being able to better stock, better look after their land. We've been at field days twice now, and nobody was shocked to see us there. And what surprised me most recently was how many Otago graduates are already there at field days. They're already out in the sector. Our graduates who have done botany and geography and marketing, they're already in the sector. We also had a great field trip last year to one of the medicinal cannabis growers down this way, which was a very quiet bus ride back. Didn't sample anything, we just had so much fun that day we wore out. More recently, talking with farmers who are looking at regenerative practices. Those who say we'll never change how we farm aren't recognising that they don't farm how their parents farmed. Farming always changes, always evolves. We find better ways, more productive ways, but we also are looking for ways that have less environmental impact, that are not only better for the environment, but are also better for the people who work or are associated with the sector as well. So I've had an absolute blast talking with people down this way who are trying to change farming practices, reduce stock numbers, go to once a day milking. It doesn't actually reduce your productivity. It can actually keep your productivity as it was, might even improve it. But you're already moving down a pathway reducing emissions. And so these people who have already gone on with this and started anticipating things or knowing we needed to change things before He Waka Noa ever came out, they're already doing it out there, and engaging with these people who then feed back into the research we can do and help them understand what is working and what isn't working has been an excellent, wonderful opportunity. I've also got to work with the first cohort of students that went through the program. What was great about a small cohort is they tend to be very honest in their opinions and understanding and in interpretations, and their feedback is far better than any student appraisal at the end of the year where they've got to write things down. When they can just tell you straight to your face, oh, we'd like to hear about this, can you arrange this speaker? You can. So with Connie and Archie and Claire are our first graduates who have done the major, and they're out there in the sector. And Archie's with Ag Research, Connie's with Fonterra, and Claire's with Silver Fern Farms. They're not on a farm every day or visiting every day, but they're somewhere in the agricultural sector. So thank you so much, and I'll finish there. Thank you.
Why the selection of Craig as the inaugural chair in agricultural innovation at Otago is so apt. Obviously, if you look back in the history, in the early 1870s, James McAndrew suggested that one of the foundation papers for a degree structure at Otago University should be agricultural chemistry. That topic was dropped for the succeeding 146 years. And it was a very long gestation, which has been completed satisfactorily 4 years ago when we had the rebirth of agricultural innovation at Otago. And obviously, this is especially appropriate as an inaugural presentation, because an inaugural presentation from an inaugural professor is about as authentic as you can get. There's only one thing worse than listening to me, and that's listening to me amplified. So we are at a special stage, and involving agricultural innovation at Otago was this very special development, which we applaud the university for supporting as a new initiative. It was exciting because we obviously had to make sure that this didn't compete with existing agricultural programs, and I think we have succeeded in going down that avenue very satisfactorily. We're living in a world where innovation, especially in agriculture, is critically important. And it's critically important because we have been so successful in developing agricultural modalities that some of them have become quite destructive. And that is where the major target for the innovation studies at Otago is looking at problems rather than looking at more production or more whatever. So this is a very special time, and obviously along the way we have caused very significant environmental incursions which have been destructive, and hopefully, I mean, we have even created a new epoch, a geologic epoch called the Anthropocene. And the Anthropocene is the first period known in human existence where what we do affects the climate and possible stability of our climate and our planet. So we have a responsibility, especially involved in university, to create an environment in which we look at solutions to these problems. So we are very excited that you have taken the chair in agricultural innovation. We believe your appointment is entirely apt because all we have to do is to look at the range of activities that you've been involved with since agriculture to make you ideally equipped to address a whole lot of quite diverse problems which are presented in agriculture. So we're very excited. If we look at the range of things you've been involved with, you've looked at formulation and delivery around nutrition, probiotics, contraception, wool healing, infection control, population control, controlled release of fertilizers, and you've target systems which range from soil to grass scrubs, from wildlife to domestic livestock, and this is the whole brief that we would expect to have incursions into agriculture at all sorts of different levels. So we're very excited. We recognize the value of applied research, and we think it's very important. You have 16 patents, you have 4 registered products that you have produced with lovely names like BioShield, Hog-On, Bait-Right, and the special cedar. Because cedars are very special in New Zealand parlance, because New Zealand has been a world leader in animal reproduction, especially in animals, and that was initially found around the cedar. So we're very interested in that. So that allows me to hopefully find the elusive prize. I see it. But this is my second duty, and that is to present you with this little token of appreciation from the university. So thank you for presenting the lecture, and we hope that will bring you some pleasure. I now have some very minor duties to perform, which are to thank the audience for visiting and engaging today. We appreciate that very much. And secondly, we would like to invite the audience for refreshments at the staff club. And for those of you not totally familiar with campus, go outside the main door, take a right turn, and keep on walking until you get up to the bridge. And that's the staff club. If you find any older person wandering around going into a building, that's likely to be somebody going to the staff club. So thank you for your attention. Thank you for your presentation. It has been a joy to hear the variety of your research. Thank you.